Hello. Let's talk about DNA replication really quick. So essentially, after this chapter, you're going to need to be able to explain how the DNA structure itself reveals its replication process and describe Messelson and Stahl's experiments. And hold on. Okay, so let's start with DNA replication, shall we? So in Watson and Crick's original paper, it does say it has not escaped our notice that the specific pairing of A and T, really Chargaff's rule, suggests a copying mechanism with specific base pairs that can be sequenced. So they hypothesized this when they came up with the structure that they stole from us and Franklin. So the double helix model suggests that the two strands separate during replication, which we know to be true, and each strand serves as a template for the next strand, which we kind of know. The question was, what was not clear is, so if you have these template strands, does the old, after they copy, does the old stay together and there's a new? Does one old stay with one new? Or is it, like a mosaic, where like some new and some old stay together? These are called conservative. This is conservative. This is semi-conservative, and this is dispersive. So, Messelson and Stahl were trying to prove which one of these it is. So again, we used E. coli, naturally, and we used some heavy isotopes. And they looked at, okay, here's the original strand, right? Here's the strand that got replicated. Now, if it replicated, you can see that it replicated semi-conservatively, right? There's one old and one new. That's semi-conservative. So when they radio-labeled it, or heavy DNA, they saw that there's one old strand and one new strand. The heavy isotope, which gets incorporated into the bases and eventually into the DNA. And you can see that there's one old strand and one new strand. And Messelson and Stahl did this experiment using heavy isotopes, right? And only half the isotope was there. So when you have this template, you get half old, half new. During this, each of the two strands that make it up were mixed with the heavy isotope, and you get the semi-conservative replication. One old, one new, each time it replicates. This is the bigger picture of that. And again, this is the Messelson and Stahl experiment. So continue with replication. We are going to just kind of get through all of replication. We're going to look at prokaryote and eukaryotics differently. Starting out strong with prokaryotes. Essentially, it's just going to replicate this circle, and it's going to keep replicating. It doesn't have to go from the nucleus. It doesn't have to do anything else. It can just replicate, and it can replicate at multiple points too. There's a replication fork, it pulls it, and it replicates. There's a bunch of structural points here, a lot of enzymes. As you can see, there's a polymerase. DNA polymerase adds nucleotides one by one. We're going to do some of this in class. We're going to actually add our little nucleotides, our A's, our T's, our C's, and our G's, and that's what the polymerase does. It adds those nucleotides one by one to the growing chain. And it's going to do this single-stranded. It's not going to do both at one time. We're going to talk about this in a minute. We're going to talk about Okazaki fragments. But right now we're going to talk about the single strand near the replication fork is going to just replicate all the time. It's going to just keep replicating. Okay, so far we have DNA polymerase, which adds the nucleotides. You also need an RNA primase. This synthesizes an RNA segment that is about 5 to 10 nucleotides long and complementary to the DNA segment. Because the sequence primes the DNA synthesis, it's called a primer. So that's something else that we need. We need a primer, and we need polymerase. Now, this is going to happen continuously on the leading strand. So the other strand, complementary to the 5' to 3'. Do you remember we counted our carbons? 5' to 3'. And this is with the replication fork. Let's talk about Okazaki fragments, because it's super fun. Okay, so the complementary strand that's 5' to 3', that's called the leading strand, and it's just going to synthesize DNA. Leading strand, it's going to go from 5' to 3'. This is where it's open, and it's just going to continuously go from 5' to 3'. Okay? Now, it can only go from 5' to 3'. So this lagging strand goes this way and then leaves these little segments, and goes this way and leaves these little segments. These are called Okazaki fragments. These little strands, and then it starts over and goes 5' to 3', goes 5' to 3', because it can only replicate 5' to 3'. Now, these adorable people are the Okazakis. Are they not the actual cutest? Look how cute they are. These are the Okazakis, married couple, who found this out, and she is still alive, crushing it. He passed away from some fallout from the Hiroshima disease, but she continued doing research and is still a valuable part of the scientific community today. So they discovered the Okazaki fragments. Again, 5' to 3', it's just continuous. The discontinuous is the Okazaki fragments. So hopefully you will be able to explain that. And in eukaryotes, it just works a little bit differently. In eukaryotes, we just have to unwind it from the chromosome, right? Because it is wound on histones, so eukaryotic DNA is bound to these histones, right? Chromosome, unwinding a chromatone, and histones, you need to unwind it from the histones, and then you have your helix. So you can't replicate anything until it is unwound. The chromatin is the complex, and it may undergo some chemical modifications, and that's what you need for replication. And we will talk a lot about that because it's very interesting in what we know about genetics and diseases, how this unwinds from the histones. So at the origin of replication, you still get the same thing. Here's our polymerase, right? Our DNA polymerase. You still have a leading strand. You still have a lagging strand. The main difference is unwinding this from the helicase. That is your main difference between prokaryotes and eukaryotes. Same thing here. Topoisomerase, which removes that coiling. You have a helicase, which unzips everything. The replication forks are the same. The openings of the DLA helix, making sure this doesn't get coiled, all the same. Again, overwinding is again resolved by this topoisomerase, and the primers are formed in almost the exact same way. And Okazaki fragments, again, this is polymerase, this is your Okazaki fragment up here, discontinuous. Okay, so that is really all of replication. We will talk about repair and some other things as we move on, but for now I really just want you to understand eukaryotic and prokaryotic replication and Okazaki fragments.